Okay, today we're going to be talking about translation. This is the next step in how do we make a protein. Originally, we started talking about transcription yesterday. Today, we're going to be talking about translation. So, introduction. This is kind of review from yesterday. First, on your notes packet, I want you to transcribe DNA to mRNA. So this is where you're using the special alphabet where the A turns into a U. So you're going to put the letter underneath this. So you're going to do these three sets of codons. So three sets of three letters. Next, what are the three bases letters of mRNA called when they are together? Hint, I just gave away the answer. Also, when I wrote up on the board. So spend the next minute or two and answer these. Moving on. So now we're going to talk about the answers. So transcribe the DNA strand into mRNA. So remember, A goes with U, T pairs up with A still, T still pairs up with A. Remember, this is the mRNA strand. G will go to C, A will go to U, C will go to G. C will go to G, C will go to G, and A will go to U. And then finally, that three-letter word is known as a codon. So the genetic code, remember, there's 4 letters in the DNA alphabet, and there's also going to be 4 letters in the RNA alphabet. So for the RNA alphabet, that's going to be A, C, G, and then U. So first you're going to have to transcribe, and then we're going to go to the translation. That is what we're focusing on now. There's 20 letters in the protein or polypeptide alphabet. I'm sorry. Polypeptide is going to be another word for how we make a protein. How do we get between the two? It is called translation. So remember, transcribing, that was where we were typing down the words or writing it down. So we're going from DNA to mRNA. Now we're going to be going from the mRNA and figuring out what the amino acids are, or the small building blocks of protein. Oops. So the genetic code. So we talked about going from mRNA to DNA. So we're going to be looking for the amino acids. So you've got those 3 letters. And the way you read this is called a codon wheel. Is we start off in the middle, and then we work our way out. So to get tyrosine, we would start off with U, which is right here. The middle letter is in the middle set of this kind of secret decoder ring, A, and then we're going to go to C. So it can be multiple letters, but here, so UAC will get us tyrosine. Tyrosine! So now it's going to be your turn to try it. Try figuring out what CAG is. I'm going to give you guys a minute. The answer is glutamine. So I would start off C, A, G, glutamine. AAA. Let me give you another minute to look at that one. Got your answer? Let's figure it out. Start off in the middle. Go to the middle ring. A, and then we're going to go A. I think it's going to be lysine. Yep, lysine. GGA. I'm going to give you a minute to look at that one. So let's start. G. And I'm going to go up to G. And then A. Glycine. Next one, GGU. I'm going to give you a minute. So now we're going to go start off G, G, U. Ooh. Glycine again. So see with this, I'm always going to have the same first letter. I'm always going to have the same middle letter, but it codes for the exact same amino acid. The amino acids are all these words on the outside. But that third letter, it can be four different letters in this case, but it'll still get me the same amino acid. Special codons. AUG, methionine, or start. The way I like to think of this is the green light at a stoplight, it means go. This means that the secret decoder ring and the amino acids, they'll start making themselves. So UAA, UAG, and UGA, these are stop codons. So these basically tell the machine to stop making everything. Stop! This acts like a period at the end of a sentence. Ricky the ribosome. So all these amino acids are assembled to make a protein at the ribosome. So remember we talked about ribosomes equal factory. We talked about this back when we were talking about cell about a month or two ago. Translation. During translation, the cells use information from the mRNA to produce proteins. Knowing which codon goes to each amino acid is the job of the tRNA. We're not going to focus too much on that in this class. tRNA. Each tRNA molecule has the following. An amino acid. So basically, it's this little thing that flies through the cytoplasm and it carries the amino acid on top. It has an anticodon, or a piece at the bottom that fits in with the mRNA. Again, we're not going to focus too much on this in this class. The anticodon bases are complementary, or means they match up with the mRNA. So when we mean complementary, like the A goes with the U goes with the A, and G goes with the C. On the ribosome, the tRNA begins binding to its appropriate spot on the mRNA, kind of like the right place where it's going to connect. So here's the ribosome, and it's going to bring in the amino acid on the tRNA, and it's going to attach, and it's going to kind of match up like with the teeth on the ribosome. The tRNA molecule binds to the next codon on the mRNA. So the tRNA with the amino acid is going to come in and start connecting, almost kind of like on the conveyor belt. So this is the mRNA code. And it's going to start connecting these amino acids. So the aspirin, the ASP, the PRO, and the methylene. And this is going to form what's called a polypeptide bond, where I like to think of like protein bond. This is get all these amino acids put together. It's going to form a nice long protein. The ribosome also breaks the first tRNA bond. So here's kind of the mRNA, and this is kind of what you've been decoding today. That tRNA is going to come in, and it's going to start assembling. See, this is like the tRNA, and this is the amino acid. They're coming into the ribosome, which is that factory, and it's going to start putting them together. And when it puts a couple of these amino acids together, it's going to form that protein. So an example was like when Mr. Franta was bleeding, and I said we were going to build new skin cells. That putting all those amino acids together is going to start getting us a protein, which could, in this case, be the replacement skin cell. Growth. The polypeptide or protein chain continues to grow until it stops, or it gets that stoplight to tell it to stop. In this case, it could be one of these 3 codons. When it reaches stop, the protein chain or polypeptide chain is released into the wild. Goes off. So think of it as like catch and release. I'm releasing the critter. 1, 2, 3, 4, 5. Let the lions go. So this is an example of the ribosome. And here's an example of the mRNA. So that kind of like the blueprint. And here's the tRNA bringing in the amino acid here. And that's going to be in the end today.